Welcome back to the Classics of Immunology Journal Club. Before we get started, don't forget to like the video on the YouTube channel so you keep the YouTube people happy. And then also click on the bell to get notified for more. Subscribe to the whole thing. Check out my website. The writing tab should have all the classic papers of immunology there, including the one I'm going to discuss today. And that paper came out in Science in 2008. The first author is Kaya Wing, who is a young postdoc from the Karolinska Institute in Sweden, who took a postdoc and went to Japan to work with Shimon Sakaguchi, who we've talked about before on this whole thing. And anyway, she did that. And this paper that was published in Science in October of 2008 is a remarkable paper. And I implore you to don't take my word for it. You got to go there and get in the weeds and read through this paper carefully. It has 4 figures, which are about 10 subfigures each. There's each different figure is an experiment in and of itself. It's probably been done multiple times. There are 13 other figures in the supplementary online material to check out if you're interested as well. And this is all about regulatory T cells. The title is CTLA-4 Control over Foxp3 Regulatory T cell Function. Now, the background for this paper is I've gone through before, but I realized I misspoke when I was talking about some of the earlier papers that in the last few weeks. This whole story starts back in 1961-62 with Jacques Miller thymectomizing newborn mice and then watching them grow up and trying to figure out whether or not that made any influence on the immune system. And of course, what he showed was, yes, it did. And for the first 4 weeks, as the mice continued to grow and they looked fine and they thrived, but they were immunodeficient, and they in the second 4 weeks of their lives, they started to undergo a GVH-like phenomenon, and they became runted, they lost weight, and they ultimately succumbed and died early with organs infiltrated with activated T cells. So that was the beginning of the whole story that became regulatory T cells. And of course, I got involved beginning in like 1981, 1982, when we first developed a receptor assay for the interleukin-2 receptor. And because one of the things that came up right away in our studies was that we had a collaborator by the name of Alan Monk, who was an endocrinologist, who told me that we really couldn't say that we were dealing with a hormone unless we could show negative feedback regulatory control over the hormone itself. A hormone would induce a negative feedback loop. We looked for that. We couldn't find it, as I've detailed, and so forth. But that always bothered me because I thought that's exactly what we're dealing with. And as time went on, the regulatory T cells became one of the negative feedback regulatory loops, and therefore very important from the standpoint of interleukin-2. As we've reviewed in the past several papers, in 1995, the people interested in co-stimulation and co-inhibition had identified in the late 80s a gene that was called CTLA-4 for cytotoxic T lymphocyte antigen 4. In 1995, they managed to knock it out of mice. The mice were born. They looked like Jacques Miller's mice. Initially, they were okay, but then they started to get really sick and runted and had GVH disease. And these mice would die within 3 weeks. I mean, this is a severe problem. And then in 1996, Maria Luisa Allegre, working in having a senior moment here, a little blockage. Anyway, Maria Luisa Allegre showed that CTLA-4 was under the regulation of interleukin-2. Then in 1996, also, it was shown that what the CTLA-4 did was that it would compete with antigen-presenting cells co-stimulatory situations by binding and downregulating B7 and on antigen presenting cells. Then also in '96, things got really hot in the '90s in this whole area. Jim Allison and his students and postdocs showed that you could increase tumor rejection by blocking CTLA-4 with monoclonal antibodies. And then, as we've talked about before, Ethan Shivak showed that Tregs, regulatory T cells that were identified by the expression of the IL-2 receptor alpha chain, if you added them to responding T cells that were trying to proliferate, that the regulatory T cells would decrease their proliferation. And he showed that you needed antigen presenting cells to be in the mix or the whole phenomenon wouldn't work. And so the question is CTLA-4 the thing that's doing that? And that's what Kaya Wing attacked in this particular paper. And don't forget that we just reviewed a year before, Pushpapandian's paper came out in Nature Immunology, showing that what these IL-2 receptor positive Tregs were doing was that they were sopping up the IL-2, so that obviously without IL-2, the cells trying to use it to proliferate couldn't proliferate. And then last week we dealt with Zoran Popmihailov's paper and myself that showed that when the cells became Foxp3 positive, they could not make cytokines, interleukin-2, particularly when you activated them through the T cell receptor. But they wouldn't be suppressive for the other population, the responding population. So the question was, is this active suppression, passive suppression with the removal of IL-2? Got to be very complicated. So that's where it brings us to the paper today. Now, in the introduction to this paper, Kaya Wing goes into the two cardinal features of T regulatory cells. One being is that they express CTLA-4 constitutively, whereas all the rest of the T cells would not express CTLA-4 unless they were activated by the T cell receptor. And then others had shown that the activation of the T cell receptor would cause the production of interleukin-2 and their response to interleukin-2 by its receptor. And that's what was regulating both Foxp3 as well as CTLA-4. The other problem that people had when they were trying to study CTLA-4 in relationship to T cells is that the T regulatory cells express the CTLA-4 molecule constitutively, whereas the responding cells, all the rest of the T cells, needed to be activated. So the question was, what's going on here with this CTLA-4 molecule? So what Kaya Wing did was that she made knockout mice where she knocked out CTLA-4 preferentially and only in the regulatory T cells, and then said, okay, so what's going to happen now? Now, the importance of this is that they made the knockout by having it be a conditional knockout regulated by the Cree gene that was under the regulation of the Foxp3 promoter, so that as the regulatory T cells were developing, really postnatally, essentially, because that had been shown in other experiments, they would then cut out the CTLA-4 gene. The other cells that weren't trying to express Foxp3 would not cut out the CTLA-4 gene. So let's go to screen sharing and see what happened. So this is figure 1. And I want to focus you down onto the D, the subfigure here, because here we have the negative control of the construct without the Cree gene. And so now we have CTLA-4 versus Foxp3, and you see CTLA-4 and Foxp3 negative cells, and you see CTLA-4 and Foxp3 positive cells. So here is the traditional knockout. So you knock out CTLA-4 out of everything, And you don't have CTLA-4 in the Foxp3 negative or the positive population. And here's the conditional knockout where you have it in the Foxp3 negative cells, but not the Foxp3 positive cells. And the bottom line is, if you do make mice like this is what happens to them in terms of their survival. Percent survival here, and we have the wild type mice that survive, 17 of them up to 80, 70, 80 days. The traditional knockout, as I said before, they went downhill and died within 3 weeks. And here's the conditional knockout. And you can see that they survived with a T1 half of about 50 days rather than about 20 days. And then by 70 days, they've all succumbed. So there's a tremendous effect of knocking CTLA-4 out of the Foxp3 population. So if we go on then to figure 2, the result of that, and the reason they die, is that they develop the typical syndrome of filling up with activated T cells. And here is the spleen, wild type, and the conditional knockout. Here are lymph node cells. And here is the heart. It's unbelievable with the negative control and then the knockout, the conditional knockout. And here are the inflammatory cells into the heart and so forth. They also develop inflammation in the lungs and and all over the body. And they develop gastritis with anti-parietal cell antibodies. They also have markedly elevated IgE, as well as IgG is not as marked. And if you look at their cytokine gene expression, here's IL-2, and here's the conditional knockout here as compared to the regular traditional knockout. Same way where the interferon gamma is elevated, interleukin-4 is elevated, interleukin-17 is only out in the traditional knockout, which is interesting. They didn't really have an explanation for that. And then if you look at figure 3, this is a tumor immunology experiment where the mice have been given leukemia cells to see what would happen if they didn't have any regulatory T cells. And here you can see that all of the negative controls that do have regulatory T cells are dying, essentially, whereas in the red, the knockouts, conditional and so forth, are rejecting their leukemia cells. So you get both autoimmunity and you get increased tumor immunity. Just want to check my notes here. So the conclusions at this stage of the game is the Treg specific CTLA-4 conditional knockout they develop and they survive normally, but they have defective regulatory functions. And that's exactly what they were they thought was going to happen and what they were hoping for. So if we stop the screen share now and go on to the discussion, they make just a couple of points, which are very important. And one is that the CTLA-4 expressed in Foxp3 positive T regulatory cells is critical for both in vivo and in vitro suppression. And it's mediated, at least in part, by CTLA-4 dependent decrease on antigen presenting cells of the co-stimulatory ligands CD80 and CD86. Because it works both in vitro as well as in vivo, they've said that therefore CTLA-4 is a key treatment target for autoimmunity, allergy, and tumor immunity. It's good for what ails you, right? And so that's the end of this paper. But subsequently, and we're going to review these papers as well. So that was 2008. 2010, Gregoire Altan Bonnet's paper came out that I was a co-author on, where he showed that there was a marked increase in the alpha chain expression on regulatory T cells. And the effect of that is that it markedly increased the affinity of the trimeric IL-2 receptor complex for interleukin-2. It was increased by at least 150-fold. And so to essentially 60 femtomolar from 10 picomolar. And that really gives the regulatory T cells a distinct advantage particularly when IL-2 is in short supply. Then Kaya Wing came back. She went back from Japan and went back to the Karolinska, started her own lab. And she came out with a paper in 2016 when she wondered what would happen if you didn't knock it out in development, CTLA-4. Because as you recall, the Tregs develop in the thymus and then populate the periphery in the first essentially few weeks of life. If you wait until the mouse grows up and becomes an adult and then knock it out, that was the question that we'll get into when we get there. So we're almost into modern times here. So don't forget to like the video, click on the bell, subscribe, check out the website, and come back next week. Thanks for listening. If you're new to immunology, or you just want to delve into a certain aspect of this large field, and you want to get a grasp on things, you should buy my new book, Molecular Immunity, because it's a chronology of all of the important experiments and discoveries that have been made in the last 60 years. It's concise, but it's also very comprehensive. And it's the only such book that's been written since 1970. So you can buy it on Amazon, and together with the YouTube videos of the Classics in Immunology Journal Club, you can't go wrong.